Hey everyone, welcome back to my channel. So in today's lecture, we'll continue with lipid rafts. So this topic is frequently asked, and once you understand it properly, questions become very easy. So lipid rafts, it is of two types, planar and caveoli. So planar contain flotillant proteins which are found in neurons, and caveoli, they are inwardly curved flask shaped rafts rich in caveolin protein. Caveolin protein are integral membrane protein. They are cholesterol binding protein and they have ability to bind with sterols. Function of lipid rafts, they are involved in signal transduction, endocytosis, cholesterol trafficking, and they also act as platform for assembly of T cell signaling machinery. So empiphatic lipid molecules form three types of aggregates or structure when added to aqueous solution in order to minimize hydrophobic contact. So these three types of structures are micelle formation, bilayer and liposome. So micelle, they form small spherical structure 20 nanometer diameter, hydrophobic fatty acid chains hidden inside the sphere. So these are hydrophobic fatty acid chains and polar heads facing outward. Bilayer, two lipid monolayers combine to form a 2D sheet. So in this bilayer, the hydrophobic region at the edges are in contact with water, which make it relatively unstable. Thus it spontaneously folds back to form liposome. So this structure forms this structure. So liposomes, they are closed, self-sealing, solvent-filled vesicles and bound by only single bilayer. Now asymmetry of lipid bilayer. So plasma membrane is asymmetrical. Two leaflets have different lipid composition. The two leaflets are cytosolic leaflet or inner layer, and the second one is non-cytosolic or extracellular leaflet, which is outer layer. So distribution of phospholipid in cytosolic and non-cytosolic leaflet. So cytosolic leaflet, it is rich in phosphatidylserine and phosphatidylethanolamine, and the function is to provide negative charge and it is important for cell signaling. Extracellular leaflet, it is rich in phosphatidylcholine and sphingomyelin, and its function is to provide structural stability and it also involved in cell recognition. So functional importance of asymmetry. So asymmetry maintains membrane stability, essential for normal cell function, and it also helps in signaling, membrane curvature and protein orientation. So now let's talk about the lipid movement or flip-flop movement. So normally lipid do not freely move between the leaflets. So three types of enzymes are involved: flippase, floppase and scramblase. Flippase, they are involved in lipid movement from outer to inner region. Floppase are involved in inner to outer region, and scramblase allow bidirectional movement of lipid molecules in the leaflets. Now clinical or biological significance. So phosphatidylserine exposure on the outer leaflet, it acts as marker of apoptosis or cell death. In platelets, phosphatidylserine exposure help in blood clot formation. So this is the important function. Motion of lipid molecules. Nature of lipid bilayer. Lipid bilayer is not rigid, it is dynamic, show fluid-like behavior. Types of lipid movement: rotational movement, lipid rotate around their long axis. Second type of movement, lateral motion or diffusion, Lipid moves sideways within the same leaflet, most common and fastest movement. Importance of lateral movement, it indicates the membrane behave like a fluid. So that's all for today. See you in the next lecture.